All right, good afternoon, everyone. Thank you for joining us. It is my honor to introduce our DYSS internal candidate. So Dr. Bahl is joining us from UW, from Molias, actually. So he received his bachelor's and master's degree at University of Maine in molecular and cellular biology and biochemistry. He then joined Dean Madden's lab at Geisel School of Medicine at Dartmouth, and during which he received 2 NRSA training grants. After finishing his doctorate, he joined David Baker's lab here, and he's been developing and using different computational methods to better understand protein structure and function and how they relate to one another, and for which he has been working under an NIH training grant during his time here. He has an outstanding publication record, including a recent Nature publication. He has 3 patents under his name and has mentored several undergrad students in his time, which he's very passionate about. So shortly, he will be leaving us at UW to build his own lab at the Institute for Protein Innovation. And so please join me in welcoming Dr. Bahl.
Well, thank you very much for the lovely introduction, and thank everyone here who I've gotten a chance to talk with today. This has been a really fun opportunity. It's a huge honor to get to present in this seminar series. Just the energy and the passion in this department is like very palpable. This is just a really fun place to be. So thank you so much for sharing this department with me and giving me the opportunity to interact with everyone here. So I'd like to share a story about what I've been working on for the past 5 years here at the Institute for Protein Design, very similarly named to where I'll be going soon. And we like to think about protein as a material, but ultimately we want to make protein do stuff that's useful for humankind. And one of the things I'm particularly interested in, and I sort of highlight up here, is the genetic encodability. So there's very few nanomaterials where the building blocks are genetically encodable, very simply, that lots of domains of life can access and use. And I think that's a power of protein as a material that hopefully will become apparent more throughout the talk. I know there are a lot of people who are very protein-centric here, and also some who are in other fields in chemical engineering. So you know, I know how the story goes, so I think dialogue is far more interesting than monologue, so please interrupt during the talk if you have any questions or something isn't clear. Discourse is really entertaining, so please interrupt. Okay, so talking about protein as a material, we really want to be able to control protein and make new shapes, sizes, and functions of proteins than what we've observed in the environment. And to do so, we need to be able to build proteins de novo, build them from scratch. So the problem space is really, I think, highlighted by this number here. So if you think about the average protein that we sequence, you could say is about 200 amino acids long. Right? You've got 20 different amino acids that could be at each position in this polymer. And so when you put this together, an average protein can have 20 to the 200 different possible sequences that it can sample. That is an enormous number, far too large for even natural selection over the face of the entire planet to possibly sample. Right? So this box is designed to make you think about the totality of sequence space that proteins can occupy. So this is fake data. But really, it's supposed to represent all sequence space, right? And these little gray circles are islands of protein sequence that we've observed from nature. And you can see that there's not very much compared to the total space. So in truth, nature has sampled a very small possible functional sequence space that this material can access. Directed evolution, where you randomize sequences starting from naturally existing proteins and then select for functionality, can push the boundaries out so you can get more functionality and you can expand the amount of sequence space that you can sample. But if you want to jump deep into these unsampled regions of sequence space, you can't start from things that we've observed in nature. You have to be able to have a new method that allows you to jump right into it. And so de novo protein design, where we're building proteins from scratch in a computer, are not restricted to the ways in which evolution works, where you have the protein is encoded by DNA, and you have to make changes at the DNA level in order to make changes at the protein level. And it's always a walk. You have to start somewhere and then make a path to go somewhere else. You can have recombination events or domain fusions that might be able to let you jump between islands, But if you want to jump right out into here, you're going to need a new tool. And so that's really our motivation for all of this, and our way of thinking about the future of engineering proteins is to access really crazy new function. You need to be able to find new sequences. So the outline of the talk is first I'll give you a story about how we build new proteins from scratch, not based off of exemplars in nature. I developed a new computational method to do this. I'll show you some experimental validation that demonstrates that we can actually do this, that it's correct. And then, when you build proteins from scratch, you can start to ask different questions than you can from when you're working with proteins that already exist. So I think we can learn some new things about protein as a material from this approach, and so I'll highlight a few of those. And then I think the thing that's more interesting that I will spend less time talking about, perhaps, is the types of problems that you can use with de novo design. And the first is taking this method to build a new platform for drug discovery, and then taking that drug discovery platform and trying to do useful things with it. And the example that I'll go over is trying to neutralize proteins from pathogens. And you can do this if you have existing information that you can steal about how to neutralize a pathogenic protein. And then I'll wrap up with a very brief vignette on my work towards doing what we consider sort of the holy grail in protein design of a one-sided interface design project. And the design approach, I should mention for this, is helpful because with design you can create a rational protein-protein interaction where you have a site-specific interaction rather than just random connections, right? So if you ever have made an antibody, you inject your protein into an animal and you're pulling out affinity reagents, but you don't get to pick where those bind. And so with a computational approach, we can start to do that. All right. So thinking about drug space, if you're thinking about therapeutic molecules, you've really got two options historically, right? You've got small molecules like aspirin over here that are very small, hundreds of Daltons. And these have some very beneficial properties, right? Small molecules, you can store them for a really long time. Chemical synthesis can be very cheap. They're orally bioavailable most often. But there's some problems with them too. You have promiscuous interactions. The drug is going to bind something that you don't want it to and have some sort of side effect. Maybe when you are metabolizing this molecule in the liver, you have a toxic byproduct that you also don't want. So there are some places where the small molecules they're not a one-size-fits-all solution. So on the other side, we have this new class of drugs, these antibodies, biologic drugs. There are some biologic drugs that aren't antibodies, but by and large, they are. Antibodies are amazing because they have a much larger surface area with which I should say down here, much larger surface area with which the affinity reagent is binding to the target, right? So with a large interaction surface, you have much higher specificity and much less off-target interactions. It's also made of protein, which is food, so there's no problems with toxic breakdown products. The problem is that the pharmacokinetics of these aren't always what you would like. You have to refrigerate them, you have to inject them to deliver them most often, and they're not very well suited for medical applications in the developing world. So there's space in the middle of this size range where you can start to grab some of the beneficial properties of small molecules and some of the beneficial properties of larger biologics and combine them into the same scaffold. And so these are constrained peptides. So peptides that actually adopt a discrete conformation. You can use these to get really high affinity interaction with your target. They're larger than small molecules, but dramatically smaller than proteins, but they have a really big interaction surface, and so you can get highly specific interactions with your target, even with a small molecule here. So, we didn't think of constrained peptides. Nature uses these for lots of things. Here's two examples. The top one is a component of scorpion venom. So this is from I think the death stalker scorpion. And this protein will bind to your ion channels in your neurons and generally cause bad things to happen. The one on the bottom here is actually from a plant. This is a trypsin inhibitor from squash seeds. And you can imagine that plants get eaten fairly frequently, and they need their seeds to be able to pass through the organism, come out the other side, and become a new plant. And there are lots of digestive enzymes in those animals, like trypsin. And in order for the seed to be able to survive, they need to have ways of not being digested. And so these peptides can also do really cool things like inhibit proteases, which are specifically designed to chew up protein. So these are two natural examples. There are quite a few others. And these conserved disulfide bonds are really critical. They give these proteins really unique properties. And the pattern of disulfide connectivity is highly conserved. It's so conserved, in fact, that it's used to define the different families of these constrained peptides. And yeah, signaling molecules or toxins. So insulin, for example, is also a disulfide-rich mini-protein. So also peptides are just very short proteins, and it's kind of a gray area of where a protein ends and a peptide begins, and where the sharp line is. So I might interchangeably use the term mini protein or peptide throughout this talk. It kind of means the same thing. So they have really interesting properties that make them exciting for potentially making drugs out of them. So they're very poorly immunogenic in general, the natural versions. There's quite a few reasons for this, and they're largely speculative. They're incredibly thermal stable, likely due to these additional covalent crosslinks that are restraining the shape of the molecule, so just removing degrees of freedom. They can often be totally protease resistant, even to the point of inhibiting proteases. You may have heard of tumor paint. It came out of Jim Olson's group at Fred Hutch. That is also a component of scorpion venom. It's another disulfide-rich mini-protein that Jim has stuck a fluorescent molecule on. And the really interesting thing about this particular natural component of venom is that it serendipitously binds to tumors, very specifically, the target we actually don't know. And it also somehow crosses the blood-brain barrier. So they can use this to light up brain tumor. So when a surgeon is inside someone's skull trying to cut out a tumor, they can make sure, A, that they get all of the tumor and don't leave anything behind, and B, that they're not cutting out regular tissue because the tumor will be glowing. So things like this are very exciting potentials that these scaffolds can give you if we can learn how to control them a bit more. I went over a little bit about the genetic encodability already in the title slide, but the one I want to drive home is that they're produced by all domains of life. So plants make them, we make them, single-celled organisms, bacteria, and single-celled eukaryotes also make them. So unlike antibodies, which are also genetically encodable but require really sophisticated cellular machinery to manufacture and secrete, these disulfide-rich peptides have the potential to go into any GMO organism. So if we're trying to think now about the bright future where chemical and biological engineering is saving the planet from ourselves, we're going to need to modify the life around us. And being able to make drugs and plug it into essentially any organism that would be useful to plug it into, I think has a lot of exciting potential for future applications. So I've built these up that they're great. So why don't we see a lot of drugs already based on them? And it's really because these peptides as a material are really difficult to work with. If you want to use a function that already exists in nature, it's really helpful. You can it already works, it's doing what you want, great. But if it doesn't do what you want it to do and you need to make it do something new, that can be really hard, right? So if you, generally how new functionality has been incorporated is through sequence diversity. So Jennifer Cochran at Stanford has probably been the most successful. She's been able to graft different loops into scaffolds that have some sort of functionality in that loop. Or perhaps you could diversify a few residue positions. Like maybe this helix could tolerate some diversification, so you could mutate each of these positions to all 20 amino acids and make a big combinatorial library and screen, right? But the amount of diversity you can get is very, very small. If you need this helix to be one turn longer or one turn shorter, it would break the whole structure in order to do that. So if you don't have a natural example, you basically can't build it. And so for the most part, there has been very, very few successes in repurposing natural disulfide-rich peptides to do something new that evolution hasn't selected for over millions of years. Making these things can also be pretty tricky, particularly the natural versions. You can imagine this would be very hard to fold in vitro. You have to get these disulfide patterns just right. And some of the organisms that make these some of the organisms that make them have whole organ systems to produce, say, something this complicated, but others might be able to make another one just straight, nothing fancy. And we don't really understand why some fold readily and some don't. And we'd like to be able to make ones that do fold readily so that they're more broadly compatible. So when I started this project, I really wanted to ask the question: can we make these? They seem hard. You have to be able to get all these disulfides right, and you have to get the structure to fold up. Is it possible? And I can spoil the story and say, yes, I probably wouldn't be here talking to you guys if couldn't do it. So now I'd like to tell you how. So I'm in the Baker lab, so we use Rosetta. It's a protein structure prediction and design toolkit. And Rosetta started with protein structure prediction, in which case you have a primary amino acid sequence, so just a string code of amino acids. And we're using the software to try to predict what the tertiary fold will look like. And at the end of the day, we don't care so much about the side chains in this. We're really trying to get the main chain, the backbone. What does the protein fold up like? And we want to know this information and only feed in the structure, only feed in the algorithm what the sequence is. More recently, the Rosetta has been readapted to do design. And design is really the inverse problem. So if we're starting, say, from a naive backbone with no amino acid identity, we want to find what primary sequence will direct the backbone to fold up in this particular way. And we care very deeply about the side chain orientation and conformation in this case. And really what we want is this primary amino acid sequence. And we do this with a sidechain-based rotamer optimization protocol, where we have a score function that takes into account simple things like solvation energy, electrostatics, steric clashing, the backbone geometry in that particular position, and sums up all those to give an energy estimate that we can then use to sample different rotamers in a Monte Carlo fashion and pick out a sequence that should direct this backbone to form the low energy conformation. All right. So I've skipped a major step, and a very important one is how do we get this? How do we get a new backbone that we haven't observed yet in nature that doesn't have a sequence? And so this was a breakthrough that came also from the Baker lab, I think in 2012 or 2013, where we figured out how to make a general method for making new proteins. It was first done in the early 2000s and then wasn't done again for a decade. And so we now have a generalized method, and this starts with blueprinting out your protein. So if we come back to our scorpion toxin example here, if we were to draw this out as a cartoon diagram so that my ape brain can start to understand how this complex macromolecule comes together, we can cartoon it fairly simply, right? So in this case, we've got 3 beta strands, which we've drawn here as a cartoon, and 1 helix, and they're connected in a particular order. And also, just to fill you in on the nomenclature, this B beta we will shorthand as an E for extended, and then the helices, alpha helices, are just an H. So when I'm showing you a topology, the order of letters is the order of the secondary structure elements in the linear sequence of the chain, and then the overall connectivity will be shown in the cartoon here, right? So in this case, the very first strand in the chain is pairing with the 3rd strand in the chain, and we can see these forming pairs here. And if we're to actually diagram this out into a blueprint that Rosetta can read, we define that we have pairs of secondary structure elements. We have strand 1 pairing with strand 3, and they're forming an anti-parallel arrangement, so we can denote that as well. And there's no register shift in between the strands, so we have a register shift of 0, right? So the exciting thing about this is we can use this blueprint information to then rebuild a new main chain, and it can be very different from what we started with. So in this case, we want to build a protein that has the same overall topology, but is totally different, right? So in this case, we've got, say, a 5-stranded, 5 residues in a strand here. Now we're building 7 residues in a strand. And we can take the information about what residue should be in what piece of secondary structure, and we know a little bit about what the backbone geometry of that position should be, right? So in the protein main chain, you've got 2 angles, the phi and the psi angles right around the central carbon, the alpha carbon, right? And different secondary structure element types will have different angles for those 2 bonds. And so there'll be a particular region of this Ramachandran space that a backbone of a residue in a helix will adopt, and likewise for beta sheets. And so we can then record this in our blueprint, and then we take a little bit of inspiration from nature. So the totality of sequence space is accessible to proteins is enormous, and the conformational flexibility that's available to the protein main chain is equally enormous. And while nature hasn't sampled anywhere close to the amount of sequence space that's available, if you look at the main chain geometry at a very small level, we believe that nature has done a pretty decent job of sampling local conformation. So if you go through the PDB and you find all the high resolution structures and you chop them up into either 1, 3, or 9 residue pieces, you could extract the main chain geometry from that and use those angles to rebuild a new backbone without sequence. So that's what we do. So we take our blueprint and we pan through backbone fragments from the protein data bank. These don't have a sequence. They're just linear main chain with phi angles that are determined by our blueprint. And we pick a few hundred of these for each residue position, and then we use a Monte Carlo sampling protocol to stitch them together until we come up with a main chain that is folded. So we use a score function in this that's it's fairly simplified. We provide energy bonuses for strands pairing together, and amino acids are represented by anonymous spheres in order to keep the secondary structure elements spaced far enough apart so that they're not packed really close together so that we can put amino acids in them later. So once we've got our new backbone, now we need to start to make it look like a protein. So the first thing we do is staple it with disulfide bonds to attempt to stabilize it. So we take each C beta atom and we draw a 5-angstrom sphere around it, and we find all the other C beta atoms in that vicinity. And then we check and see, is the geometry between these two atoms consistent with a disulfide bond? If it is, we record it, and then we can make a table of all possible disulfide connectivities that backbone can hold, and we can rank order them based on a rough estimate that we figured out on the degrees of freedom that the main chain could sample that are now frozen out by the incorporation of that disulfide. So we call it a disulfide entropy reduction. So it's an estimate of how stabilizing each disulfide is. In effect, it's spacing the disulfides out through the backbone, and then we can rank order all the possible connectivities of disulfide bonds in the main chain based on this estimate. And we pick generally the top, say, 20 to then go and design further. So then the final step, once we have our main chain with disulfides stabilizing it, is to find amino acid identity for every other position. So we use the same rotamer-based sidechain optimization protocol. And we know a little bit about what these amino acids should look like. So on the surface, we want polar residues. So if the sidechain is pointing towards the solvent. If the sidechain is pointing towards the interior of the protein, we call this a core, and we want it to be aliphatic, hydrophobic residues only. And then at the interface between the surface residues and the core residues, we call the boundary. And then this is a place where kind of anything goes, and we allow the score function to guide the selection of each amino acid in that region. And so what we're trying to do is define and design idealized proteins. So these are as perfect as protein could get. And so we've got only hydrophilic residues on the surface and only hydrophobic in the core. And this is not really how proteins we observe in the environment look. Generally, there's some mixture, and often that's involved in function. But in this case, we're trying to just make ideal proteins for now. Right. So then, so once we have this model, we really have a full atomistic representation of this peptide down to every last hydrogen. And we can extract the primary amino acid sequence from this. And our last step before we order a synthetic gene and try to make this in the real world is to see what it would fold up like in an independent folding simulation. So we use the Rosetta ab initio structure prediction algorithms to assess what the sequence will fold up like. So in this case, each dot on this graph is the result of an independent folding simulation. On the y-axis is the energy of the system. And on the x-axis here is how different that conformation is from what we designed. So 0 would be the designed model. These orange boxes here are, we start the simulation from the designed model. So and then you can think of it as a simulated annealing run. So we're trying to see if the new protein that we made is in a really constrained shape, and would it want to blow open if we allow it to wiggle a little bit. So it should stay clustered around its initial conformation, which is what we see here. And then each of these dots is a folding simulation that starts from an extended chain, and again uses a Monte Carlo-based fragment insertion protocol in order to try to fold this up into a low-energy conformation. And the these folding simulations very rarely find the lowest energy conformation of that sequence. But what it does do is find the energy of lots of different conformations. And this is actually exactly what we want. We want to see what the energy landscape of this peptide is. What are the different conformations that it can adopt, and what are the energy of each of those conformations? And what we want to see is that as the conformation gets closer to what we designed, that the energy of the system drops. So we call this a folding funnel. And so you want to see a nice folding funnel that the primary amino acid sequence that we designed is directing the peptide to fold up into one specific conformation.
Can I ask a quick question?
Please.
How do you handle kinetic traps?
Kinetic traps. Well, yeah. What we would hope to see is if there is an alternative conformation that is, say, if you had a peak over here, right, that maybe wasn't as deep into a well as here, you would be able to see an alternate conformation in this energy landscape, and maybe we wouldn't select that design out of fear that it would get trapped into an alternate conformation. Yeah, exactly. Yeah, it's a good question. All right. So I told you how we make this scorpion toxin topology, and there's these 4 secondary structure elements, but really I wanted to make a method that had broad applicability that you could use to make proteins with lots of different sizes and shapes and topologies. So the first thing I did was take these same 4 secondary structure elements and recombine them in every possible way. So there's 4 ways that you can combine these 4 elements. And then wanted to move a little bit further away and try to do something a bit different. So if you drop one of these strands, now you can make just 2 strands and 1 helix. You can keep an anti-parallel beta sheet like this, or this one I think is probably my favorite, is you can start to make parallel sheet designs. And these are a bit unique. Nature very rarely has a parallel beta sheet where both edges are exposed to solvent. So this is a little interesting. And then wanted to see what we could make with just one type of secondary structure element. So of course the classical helical bundles, so just 2 or 3 helices. And then I spent quite a bit of time trying to think about how I could make something that only had beta strands and still be really small. And one of the simplest topologies I could think of is a sandwich. So in this case, we have 2 3-stranded beta sheets that come together like a sandwich and then have a disulfide delicious lunch meat inside. And this still managed to be fairly small. So all of these designs are under 50 amino acids. Okay. So I mentioned that disulfide-rich proteins can be difficult to make, and it's quite true. I think some people in the room here also have some experience with how difficult this can be. I tried some off-the-shelf stuff like PET32 and origami cells, and none of that really worked quite very well for me, and it was really tough. And in order to make this project work, I knew I was going to need to screen lots and lots of things. And so I needed a really robust protocol that could be used to screen lots of different proteins. And one of the ways I figured I could get around that was by using a fusion. So the fusion would carry a lot of the expression and solubility properties of the whole molecule, and then you could kind of drag something along with it. So even if it was a bad design or a peptide that wasn't folded as we intended, we'd still be able to purify it and test it and actually see that it was unstructured. So this fusion is actually in Adgene, if anyone wants to grab it. So it relies on this OSMY protein. It's originally discovered because it was osmotically inducible, although I think later studies have shown that it's not. But regardless, it's very abundantly secreted, so it has this sec tag on the front that's removed. It doesn't seem to have any function that we know of. Then there's just a polyhistidine tag for immobilized metal affinity chromatography, so we can purify these things easily. And then I included this SUMO protein. So SUMO is it's a ubiquitin homolog. It's in all eukaryotes. It's really nice as a fusion partner because it helps to chaperone the folding of things that are directly adjacent to it. And one of the nice things is that there's a protease that specifically recognizes the folded SUMO protein in cuts, and they can do so with scarless cleavage. So then you can make a little peptide and cut it off this big fusion protein, and you can have it without any additional residues. Right? A lot of proteases will leave 3, 4, 5 residues of scar, and if you have a 40 residue protein, then 10% of your molecule is scar now. I didn't want that. So this system worked quite well. SUMO is the protease as great as it is, also has some drawbacks. It's a part of a big molecule. The one most people use in the lab is from Saccharomyces cerevisiae, and they've hacked part of the protein off, and you need detergent, and it's unstable. I was having some trouble with it, so as a little side project with some really talented undergrads, we redesigned the protease and made it totally thermal stable. It's now a rock. So that work should be published soon, but if anyone is interested in trying this out, it's all internal at UW, so you're welcome. I'm happy to give you the constructs right away. So the nice thing is, because this is secreted, and the reason why I wanted to go secreted is because disulfide bonds don't form inside of the cell, right? And in the E. coli periplasm, you've got lots of disulfide chaperone machinery, which I wanted to access. And so this will direct it out, and it also helps with purification. So you can get this protein out of the periplasm, or if you express it really strong and you add a little detergent into your cultures to make the bugs a little bit sick, you can get this protein to go all the way out of the cell. And if you need to screen 100 different proteins, you don't want to have to crack open each one of those cells cultures. It can be quite laborious. So this works quite well. This gel doesn't show up terribly great, but you can purify the fusion protein. If you add the protease, you can see a reduced size of the fusion, and then you have a new band showing up, which is our peptide. And a really fantastic diagnostic it's so simple and beautiful, is that if you have a peptide running on one band on a gel. I should also say I didn't come up with this. This was our collaborators at Fred Hutch who told me about this, and I'm very thankful for that. If you have your peptide forming one discrete band on a gel that then is redox sensitive in its migration, so if you add a reducing agent and break apart the disulfide bonds, it changes its migration. And if you have one species in each redox condition, it's really strongly indicative that you've got one stable conformation that is dependent upon disulfide bonds. And so this was one way we screened through lots and lots of designs. Okay. So these 9 topologies, here's one example structure designed model for each of these. I originally designed thousands of peptides in the computer, selected a much smaller number to actually test and try to make in the real world. I think all in all, tested about 130 different peptides, had a success rate a little over 20%. And then I selected one representative peptide for each topology for a really in-depth characterization and analysis. And that representative peptide is shown here. So each of these has a energy landscape that directs the backbone to adopt the one stable conformation. So I could purify all of these, and circular dichirism spectroscopy is actually a really helpful technique for dealing with these designed peptides. So the native ones that are all kind of loops, CD is not so useful, but if you have a lot of regular secondary structure, it can be really powerful, right? So CD spec, maybe for those who aren't familiar with looking at these curves all day long, the different secondary structure elements give characteristic signatures on the trace. So you have strong negative peaks at 208 and 222 nanometers when you've got helical structural components in the solution. And if you've got beta character, you have a strong peak at about 218 nanometers. And if you've got a topology that has both pieces of secondary structure, you get a plateau across that space, right? So the CD traces show that each of these topologies is folded and has, well, I shouldn't say it's folded, shows that it has the secondary structure elements that are designed. And in black here is the line at room temperature, and if you heat the solution up to near boiling, you get this red line, and you can see that it moves just a little bit, which essentially means the protein's not really changing shape. And if you cool it back down, then you readopt the initial conformation. And if you blast the disulfides open with a strong reductant like TSEP, now you adopt this random coil. So if the line just plunges down, that's indicative of random coil. So that means that the structure is dependent upon disulfide bonds. And while some of these are very, very stable, some of them will actually flex quite a bit when you heat it, and perhaps retain some of the secondary structure character. So that means that the helical propensity is locally encoded is very strong, so that even after you break the protein apart, you still have some helical character. So the nice thing is you have the potential to tune properties in these. If you want something that really is a brick, you can do that. If you want something that might flex, you can do that too. In one case, one of these peptides even fell apart at higher temperature and didn't refold. So you really have a lot of control. I think it's quite a benefit. You can get a peptide that does exactly what you want it to. We also checked for chemical denaturation. So in this case, we did a titration all the way up to 6 molar guanidinium. So this is a harsh protein denaturant, and this is as high of a concentration as you can get. And in many cases, you can see that the protein is totally unchanged. Its secondary structure has not flexed at all, even in the presence of saturating amounts of guanidinium hydrochloride. So these things can be super duper strong. But if we don't want that, we can also titrate these to get a lower stability. So you can tune the properties to be exactly what you would like. So the CD is strongly indicative of these peptides being designed correctly, but it only gives you resolution of what secondary structure is there, not how these elements are arranged. So the true proof is to actually determine structures of these molecules. So we set out to do this. This was a collaboration with 3 different structural biology groups over the course of about 4 years. And we were able to solve the structure of 7 of the 9 peptides. So in each case, the design model is shown in rainbow, and the experimentally determined models are in gray. So this one structure is a X-ray crystal structure. There were 3 copies in the asymmetric unit, and the main chains are all aligned here. In each case, the others are determined by solution NMR, and the ensembles are also shown aligned. In each case, the all-atom RMSD was 1.5 angstrom or less, and here I've shown the lowest energy experimentally determined model aligned with the design model. And you can see that the core rotamers have a high degree of accuracy. So not only can we design the main chain to adopt the fold we want, we can even specify the rotamers of the residues in the core. So I feel confident in saying that we really have true atomistic precision over these really large, complex macromolecules. And we can control the size, we can control the shape, we can control their stability, and we can even place the core residues exactly how we want them. So now we've got figured out how to make them, and I really wanted to take a step back and think about now what sort of takeaway messages can we glean from this process? What new things can we learn? And I previously discussed how conserved these disulfide patterns are, and this had led some researchers to speculate that the pattern of disulfides was critical for folding the molecule, that there was a specific order in which these disulfides would connect, that even perhaps there was some exchange that was going on during the folding process, and that the disulfides were critical for this molecule to fold. How could they not be? They're so conserved, right? Well, that may be true in some cases, but it's not required. So these 3 designs all worked, and the disulfide patterns are all widely different, yet the overall topology is exactly the same as a natural exemplar. It seems to be that from a materials perspective, in order to have one of these fold up, the disulfides just have to be in a place where the main chain geometry is compatible with disulfide formation. That's all you need. It seems to me, my hypothesis is that the reason why these are so highly conserved is because of the restrictions of the genetic encoding of these materials. So if you want to put this disulfide somewhere else, right, there's only 2 cysteine codons, so you're going to need to make mutations to break this, probably more than 1 nucleic acid change. And then you'll have to reform a disulfide somewhere else in the structure. So you've got another perhaps 2 to 3 mutations that need to occur at each site. And that's a lot of non-functional intermediates to pass through. If you break these disulfides, even one of them in a natural protein, it falls apart. It doesn't do its function anymore. So evolution is not very tolerant to non-functional intermediates. And if you have to pass through, say, 4 of them in order to get a new functional molecule, and that's even if you somehow can stack all these up in a nice linear path where it doesn't have to sample other non-functional sites, that's still too much. So I think these disulfide patterns are so conserved because that's what nature found first, and it's just too hard to find something new. So I think that also, just to drive home the point of the design is really exciting because we're not restricted by these arbitrary limitations of a genetic encoding. We can find functional spaces in this material that evolution is physically restricted from sampling. Another interesting thing, and perhaps this seems obvious in hindsight, but I think maybe a lot of science does, is that what I started forcing there to be at least one aliphatic residue on each secondary structure element that would contribute to the core of the peptide and direct the folding, my success rate shot through the roof. And I didn't necessarily require that in the beginning because nature doesn't always do that. A lot of these peptides don't have an aliphatic residue on each piece of secondary structure directing to the core. This one clearly doesn't. So I didn't think that was necessary. And it isn't always, and we can do this without, but it's much more likely to be successful when it is. And I also mentioned how the natural versions, you can't make that many mutations to them. The structure and the sequence are really closely intertwined. And if you don't have a hydrophobic core directing the folding, it's easy to understand why that might be. You can have a lot of electrostatic interactions, and charge residues on the surface can now be involved in folding these natural peptides. But if you have a hydrophobic core that directs folding like a regular protein, now you can start to have much more diversity on the exterior of this molecule, and the interior will direct the folding. And you can start to incorporate exciting new function on these as a more generally useful platform. So in the last few minutes, so I'm running a little bit late, would like to try to just show our current efforts into functionalizing these things and actually doing stuff with it. So the high throughput drug platform, I think, is quite exciting. And one of the things I was a bit dismayed to learn when I joined the Baker lab in 2012 was how much art was involved. Every design that came out of Rosetta was looked at by a person by eye, and they made mutations by hand in, say, Folded, a video game, right? And so like this was the state of the art. People were looking at everything and using human intuition to make changes. I feel like it's very millennial to this, artisanal proteins created with a renewable trainee resource. So I knew that if I was going to be successful, I'm an engineer, not an artist. I respect art, but I know that I can't do it. So I knew that if I was going to be successful, I needed to bring engineering into this, and I needed to make it automated. I needed to remove the art from this. And this was really a new concept in the Rosetta community, and I'm very happy to say that today it's much more common, and this is kind of the gold standard, is that Rosetta does the design, and the human intuition is captured in the protocol development, not in the product evaluation. So in this case, just to really walk you through, briefly, this is all, and we published this last October, and it's all in the supplemental material. I wanted this to be automated so we can scale it and use it for lots of things. And I also don't want this to be a specialized tool that only one lab gets to use. The purpose of building this is to make it available for other people to use and build on and change. And other people need to be able to run it for that. You can download the scripts and just run it on your desktop. We'll assign that secondary structure bonus to make the strands pair together. We'll build the backbone for you, incorporate disulfides, and design the rest of the sequence. And then it will filter and throw out bad stuff to make sure that you've got those secondary structure elements on every core, that you've got actually the number of secondary structure elements you were trying to make, and that the disulfides are all of sufficient quality. And so I turned this crank on my desktop computer, and in an afternoon created this design. I assessed the folding of it, and you've got this nice folding funnel that causes it to fold up to the design state. So anybody can run and do this now. And we can also turn this crank in high throughput and make thousands and thousands and thousands of these and start to design them to do useful things. And where this, I think, the power of this is when you start to incorporate this technology with existing high throughput methods. So microarrays were a very revolutionary technology in the early 2000s and have kind of been supplanted by RNA-seq. But the core piece of technology that made that happen was the ability to print DNA oligonucleotides on a CMOS chip, right? So the DNA microarrays, they would take these stretches of linear single-stranded DNA and print it on these chips and then use that to hybridize. Well, if you cleave those off, now you can have an oligo pool where each piece of DNA is intelligently designed. These aren't random sequences. Each one is a discrete printed sequence. And you can get, say, 27,000 of these all in one library. Well, these peptides are small enough that you can get the whole gene on a single oligonucleotide. So now we have access to truly enormous amounts of design throughput that was, say, 5 years ago, a complete fantasy. And you can combine this with a technique like yeast display, where you can take your thousands of genes, add them to yeast. The yeast will piece the genomic elements together for you, and then we use a genetic fusion that displays our peptide on the surface of the cell. We can label the cells that are displaying a peptide with a fluorescent antibody, so we can light up all the cells that have taken a piece of DNA, incorporated it, and displayed it on the surface. And if we're trying to engineer, say, a specific protein-protein interaction, we can fluorescently label our target protein, and cells that glow with the fluorescent label from the target will now glow a second color. So if the cell glows with 2 colors, it's displaying and it's binding, we can fish this out with sample flow cytometry and then identify the cells that contain a successful design using deep sequencing. I'm really running out of time, so this was kind of a crazy idea, and I managed to convince David Baker and Aaron Chevalier, who was then a graduate student, now a postdoc, very talented guy, to try this out. This was really new. No one had ever even thought of doing something like this before with designs. And luckily, Aaron said yes, and we recruited 2 other postdocs. And so this is work that these 3 talented postdocs really spearheaded and I helped with, that shows that we can functionalize these. So I guess just to really go fast here, the lowest hanging fruit for functionalizing these is to take a therapeutic target, say subprotein that a pathogen uses during infection. And if you know that if you can bind that target in one specific site, you can inhibit the pathogen. Those are great targets, particularly if there's some structural information on how to do this already, and especially if that structural partner isn't very useful for a therapeutic. So we picked influenza hemagglutinin and botulinum neurotoxin B for the initial variants. We have existing structural information on what a small fragment bound to that specific functional site would look like. And it doesn't give you a huge amount of surface area when you have just that isolated fragment. So we built libraries of larger peptides with disulfide bonds that could hold this and then create additional interface interactions. So you can see our initial docked peptides were quite small, around 1,000 angstrom, and you can get a much larger surface area if you stabilize it in a peptide. We can get these oligo pools and use next-gen sequencing and fish out things. And just to punchline this, it works. So here is influenza hemagglutinin and botulinum neurotoxin B. The initial design dock here shown in green, and then magenta, and in orange here is the experimentally verified model. So we can get them to bind in a site-specific way. We can build the topology and still have it fold up properly, even when it's got an interface on it. These interfaces look great, and the peptides are also stable. And they also work in animals. So this is checking to see if the influenza neutralizing peptide will work. We showed that it worked as a prophylactic. We also wanted to see if it works as a drug after the fact. So we took mice and gave them a lethal dose of influenza intranasally. And you can see the control mice die all in a week. So these mice just received bulk protein, just BSA, as a negative control. And so all those mice died. And we checked how sick they were by monitoring the weight loss. So when the mice lost 30% of their body mass, they were euthanized. And after the lethal challenge, they just get sicker every day until they're euthanized. And then we wanted to see if this could come in as a therapeutic intervention. We need to be able to dose our inhibitory peptide after exposure. So we went in 1, 2, or 3 days after and delivered the drug. And you can see when we delivered this neutralizing peptide, the mice immediately stabilize and then recover. And so all the mice live after they're given a dose of our neutralizing peptide. So it's quite exciting. And when these peptides were delivered intranasally, they also elicited no immune response that we could detect. So it's quite exciting. I think this is a platform that we can use to make new drugs. And in the last -2 minutes, I'll tell you a little bit about this holy grail of doing interface design without having a pre-existing fragment, where we can start to tackle new challenges that we haven't seen before. And in this case, I'm picking a bacterial enterotoxin, so targets in the gut where biologic drugs are really going to be useless, and small molecule drugs are also potentially poorly suited. Some bacteria have toxins called superantigens that work by clustering immune receptors and causing the immune system to go haywire. And it does this by mediating protein-protein interactions that your immune system doesn't want to happen. And so just to summarize this very briefly, you can imagine a very simple therapeutic regime where all you have to do is break up this complex. So we have our toxin that's bringing these two immune receptors together. If you can disrupt this interaction by binding to the toxin in the same place, now you can disrupt this interaction. So I took a large library of designed peptides, docked them against the surface of the toxin in a specific place, designed the interface, ordered a large library, screened by surface display, picked out binders that looked like they were binding, did affinity maturation, and have some results. So this is work done with an incredibly talented graduate student in the lab, Cassie Bryan. She was the one manning the cell sorter. So we've got two parent scaffolds, this 1047 and 913. And so in this case, the x-axis is the fluorescent signal for display. Each of these dots is a single cell. And on the y-axis is the fluorescent signal associated with binding. And so in our negative control in red here, we have a population of cells that neither display nor bind. They're just kind of hanging around. We have another population of cells that does that does display. And when we add in our toxin molecule at either 1 or 5 micromolar, we can see a shift up. This is fairly weak binding, but luckily the native interaction between the toxin and the target is actually also low micromolar, so it's a nice target because it's quite competible. And this population of cells are displaying a designed peptide and binding it to the target. So this is what one of these looks like, this parent 913. You can see there's a great shape complementarity between the peptide and the surface of the molecule, so we can really shape our peptide to the surface that we want to interact with. And I've been able to purify one of these, and it also gives the sort of classic thermostable CD trace that's indicative of this topology. So these data are kind of hot off the press, so unfortunately I don't have any punchline for this for you, but I think we're making progress on our big challenge, and soon we'll be able to design peptides to really bind any surface that we want and exhibit some sort of function. So just to wrap up briefly, I've first told you a little vignette about how we can build new proteins from scratch, new peptides, and we can get them to form user-defined topologies and shapes, and we can do so with atomistic precision. And these peptides we can also incorporate function into them by changing the surface and get them to create site-specific affinity reagents. And with that, I'd like to thank everyone here for your attention and discussions today. There's so much collaboration that went involved in this, so yeah, thank you all. Yeah.